In this video, we are going to talk about viruses and multiplication process. So, as recall from the previous video, viruses are acellular, very simple particles that are not able to replicate on their own. So how do they multiply? They're going to have to take over their host cells, and they're going to have to use the mechanics of the host cell in order to synthesize proteins, assemble it in order to make more viruses, which can then go out and infect more cells. The process that occurs in the eukaryotic cell is different from that of the bacteriophage and what happens in the prokaryotic cell. So we're going to review both of those in detail. You do need to pay attention to the similarities and the differences in the processes. So this is the overview of the animal virus from entry to release. Let's go step by step here, starting with the first step, which is attachment or absorption. So the first thing to recognize is here is our virus, and here's our host cell, and inside here with the cytoplasm. Notice that in order for the viral process to occur, for recognition, to ensure that the virus has the right host, a protein on the virus has to recognize and attach to a surface protein or a surface receptor on its host cell, and that's what initiates and begins the process. So the virus is going to bind to a specific receptor on the host cell, and that is step 1, referred to as attachment or absorption. Step 2 is referred to as penetration because it's when the virus enters or penetrates the cell and it has to go through that host cell membrane. And there's 2 different methods this can occur. So here we are down here in our general thing. We've got to go from attachment. Now we have to get the virus across the cell membrane. That may occur through fusion. And in direct fusion, once you have this attachment, this is going to be in an envelope cell. So you see here we have the envelope. It fuses with the cell membrane, and then it just pushes everything, kind of flips it inside out, if you will, pushing all the contents of that virus into the inside of the cell. So here's where the cytoplasm and inside. The second method is through endocytosis, and this is going to be one that once the virus attaches to the surface receptors on the host cell, that triggers this host cell then to engulf, pull that viral particle in and form it in its own little vacuole, if you will, its own little vesicle. And then through the enzymatic processes here, then it's going to then cause that virus to lose its envelope, and it's going to push the contents of that virus into the cytoplasm or into the inside of the cell. So in both cases, you're pushing the contents, that nucleocapsid, into the inside of the cell. In one case, this one down here, the whole virus comes inside, envelope and all, if it has an envelope. Up here, this is going to occur with envelope viruses only, and you're going to push the capsid inside. So then once it gets inside, so now you see we're at step 3, then it's the uncoating process, because what do we have to uncoat? We have to get to the genetic material. So notice here in the picture, the genetic material is intact, but we are losing that nucleocapsid, or excuse me, the capsid part of the nucleocapsid. So you can see how it is disintegrating into each individual capsamere. Now, because the cell comes in 2 different ways, the process of uncoating occurs in 2 different ways. So I'm kind of flipping between the slides back and forth here. So again, just like we brought in the virus through direct fusion and endocytosis, we're going to uncoat the process. In both cases of uncoating, what it is the host enzymes go in and they will then start to digest the proteins that make up that capsid. So in the case of, let me go back up to this slide. So in the case of fusion, where you just push that nucleocapsid into the cytoplasm itself, then it's going to be in the cytoplasm for which there's going to be those enzymes here that are going to digest. In the case of endocytosis, where remember the whole virus is then encapsulated in a vesicle within the cytoplasm, so then that's going to be where the enzymes are located, are going to be in that vesicle, and then they're going to tear down that capsid. So the end result from step 3 is that we've digested the protein capsid and freed up the genetic material. So once we've freed up the genetic material, now the virus has to take over the host cell mechanics and synthesize and which means to make those proteins needed. So it's going to have to make the viral proteins, the capsomere, and it's going to have to replicate its genetic information. So that's the synthetic phase, and then it's going to have to put them together to form new virion, because we're going to get them outside. So the new virion here, and that's going to look like this. Okay. So here is 4 and 5. So we have gone through synthesis, and then we're going to go through assembly. So we have to synthesize the proteins for the capsomere, and we have to then replicate the genetic material. So that is what the synthesis is for 4 and 5. So you're going to be making proteins and duplicating or replicating genetic material, and then 6 you're going to be assembling it to make more of those nucleocapsid components. So let's go one more slide and look a little bit closer here at this, because it is going to vary between DNA and RNA viruses. I know you're thinking, well, it's already varying between animal and bacteria. Yep, but now in animal viruses, because think about where DNA is kept in a eukaryotic host cell. DNA is kept in the nucleus. RNA is in the cytoplasm. So the synthesis and assembly are going to vary between DNA and RNA viruses. So assume then where we're picking up with this one is number 1 is right here. Okay, so you're bringing it in at step 4. So 1 and 2 are 4 and 5 basically. So if it is an RNA virus, the majority of the synthesis is going to occur in the cytoplasm. If it is a DNA virus, most of the synthesis is going to occur in the nucleus. Now, the protein synthesis, as you may recall from the last chapter, protein synthesis occurs within the ribosomes, and those are going to be found on the rough ER and out in the cytoplasm. So they're going to be found outside of the nucleus for the most part. So in that DNA virus, those capsomeres, those proteins needed for the capsid, is going to still occur in that, as you can see here, kind of the rough ER component. So you don't have to know all the steps, but what I do need you to recognize is that in an RNA virus, when it gets into the host cell, its synthesis and replication of genetic material is going to occur in the cytoplasm, whereas the DNA virus, the replication of its genetic material is going to occur in the nucleus, and then that capsomeres, its proteins, are going to be occurring in the ribosomes as well. So that is steps 4, 5, and then 6 out here in the cytoplasm, either way, through RNA or DNA. And here you see RNA is represented in blue and DNA is in red. But as you can see, in either case, assembly, step 6, occurs in the cytoplasm. Okay, so again, when you talk about the synthesis phase, where we are making the proteins for the capsid and you're duplicating the genetic material, in the DNA, that's going to occur in the nucleus. In the RNA, it's going to occur in the cytoplasm. The assembly, where we put it all together, in both cases is going to occur in the cytoplasm. So now we have gotten the virus into the cell. We've broken down and freed up the genetic material. The virus has taken over the host. It has synthesized proteins needed for its capsid. It has replicated its genetic material. It has reassembled everything into a nucleocapsid. So now what? It's got to release. In order for it to continue to do harm or to continue to be able to replicate, to multiply, it's going to have to release from one host cell and go to other host cells. So now we're going to release those virion, and there's 2 methods for release: lysis and budding. In lysis, you're going to burst open the cell membrane, so it's literally going to rupture the cell membrane, and it's going to actually kill the host cell, but you're going to release large numbers of viruses all at once, such as the poliovirus. Or then you're going to have a budding process, and in the budding process, the individual virion are going to push their way through the membrane of the host cell and do that one at a time, budding, so they don't kill their host cell. The process looks like this. So here is the case of lysis. In the lysis, in this example, you can see that the release is of a naked virion because there's going to be no envelope around it. And you can see how the virus has literally put a hole in, bust open through that host cell membrane. In this one down here, it's called budding, and so here the virion has pushed its way through and it's taken on a piece of the cell membrane, and so this will then become an enveloped virus. Now recognize that it's going to be either this or this, even though I have them both pictured on the same cell. It will not occur both ways. It's going to be one or the other, but I put them in together just for comparison. So looking at the whole process again very quickly. So the first one is you have to attach. You're going to use the proteins on the virus is going to attach to surface receptors on the host cell, and that's then going to initiate the process to get it inside. Now, step 2 has two options. It can either push its way in through endocytosis, and it will then get engulfed as a vesicle and get inside the cell, or if it's an enveloped virus, it may just fuse with the cell membrane and push the capsid inside. But step 2 is to get it from outside to inside. Once it gets inside, then step 3 is the uncoating. And by uncoating, we mean that it's going to take the capsid and remove it from being around the genetic material. And that's going to be happening from enzymes provided by the host cell. So we're going to release the genetic material in uncoating step 3. Then step 4 and step 5, this is the synthesizing proteins and replicating the genetic material. Synthesizing proteins is going to occur in the ribosomes, but the replication of the genetic material will vary. If it is an RNA virus, that's going to occur in the cytoplasm. If it's a DNA virus, it's going to occur in the nucleus. But once we finish synthesizing the capsomere proteins and to make the capsid and then to replicate the genetic material, then step 6 was assembly, and you had to put those together. Once you put them together, then they have to get out, and step 7 is the step of release. And just like there's two processes to get it in, there are two processes that can occur to get it out. If it is a naked virus, then it lyses the whole cell and a bunch get released at one time because you put holes in the cell membrane and the cell literally explodes, releasing all these virion. If it is a budding process, such as what occurred here in this picture, then the virion pushes up against the cell membrane and pushes itself through one at a time, keeping the host cell intact, but in the process it puts an envelope around it and it becomes an enveloped virus. So that is the animal virus replication process. A similar process, but it has some uniqueness, occurs in the prokaryotic cells with the viruses, and this is what we refer to as a bacteriophage. So a bacteriophage is a term you need to know. It's a virus that infects the prokaryotic cells or the bacterial cells. The bacterial cells that get infected can have similarities, but it's also going to have some major differences from the animal viruses. Instead of 7 steps, there's going to be 6 steps, and this is just a visual of the steps in totality, if you will. So the first step here is what we call attachment or absorption. Again, remember that's the same term we used for an animal virus. So what's going to happen is you're going to have proteins. Now let me point out here, I'm going to stop for just a moment. You see how this virus looks very unique and weird? This is what we call a complex virus. So a very unusual, strange shape it may have, and that is what this one is. But these tails are spikes or proteins. Notice here, let me orient you, this is the bacterial cell, so this is going to be that bacterial cell envelope. And so the spikes or the proteins on the virus are going to attach to the bacterial cell, and that recognizes it has a cell for which it can take over and become a parasite with. So that's attachment. That's very similar steps that occur as with the animal cell and the eukaryotic cell invasion. What is different here is penetration doesn't have two possibilities as it does with the animal or the eukaryotic virus. Here it's just going to have, as you notice, it's almost like a syringe where you're just injecting a piece, but the syringe and the needle never actually go in and stay permanently within the body. Same thing occurs here. So in penetration, the tail fibers have attached, and then the capsid portion just pushes down like a syringe, and notice what happens is it pushes the genetic material straight into the bacterial cell. So then you've got to go through the next step, and I'm going to skip the slide there, but we will get back to it. So once you go back in and you have that bacterial genetic material in there, so notice we've penetrated. Now you got to go to duplication, assembly, maturation, and release. Notice the step that's missing, uncoding. Why don't you have to do the uncoding as you have to do with the animal virus? Because again, it's just pushing it straight in there, and the capsid never enters into that bacterial cell. So if the protein capsid never enters inside, then you don't have to do the step of uncoding. So once you get that bacterial genetic material into the bacteria, once you get the viral genetic material into the bacteria, there is a scenario which can occur. In the animal cell, for the most part, they're always just going to immediately become active, go through that process of synthesis, etc. But in the bacteriophage, in the virus that infects the bacteria, there is two different options. It can go through a lytic stage, which is the active stage, where it immediately goes through the steps of duplication, assembly, release. And then there's a lysogenic stage, which is a latent or temperate phase. And in this case, notice here, see the red? The red is representing the virus genetic material. The virus genetic material in the lysogenic phase gets inserted into the bacterial DNA, and it just sits there. So every time the bacterial cell replicates, it replicates its entire DNA, and as it replicates, it takes that viral DNA with it. And so each and every new bacterial cell is going to have the viral DNA with it until the conditions change. And then once the conditions change, then it all of a sudden induces that viral DNA to become active, and then it forces it from the lysogenic stage into the lytic stage. So you go from attachment to penetration, and then that bacterial virus has to make a decision. Is it going to become active, or is it going to go into the latent or dormant phase? So let's assume it's going to go into the active phase and go through the next three steps. So duplication. Here in the duplication, the bacteriophage has to do just like we did in the animal virus. It's going to have to use the host cell's mechanisms to duplicate its genetic information. Then it's going to have to also create the proteins needed for all of the pieces for that bacterial phage. Then it's going to have to assemble, and because of how complex these viruses are, it has to not only assemble, but then it has to add pieces to it. So it has to mature, and then it releases. So let's look at these 4 steps in a little bit more detail. So here's the viral DNA. First thing it's going to do is come through here, and notice it's duplicating. It's taking over the bacterial's mechanisms, and it's making more proteins, and it's going to duplicate its genetic material. Then it's going to assemble, and when it assembles, it's actually putting the genetic material inside the protein capsid, and it's forming the nucleocapsid. But then maturity has to occur. Why? Because it's got to make its sheath and its tail fibers. And so then it has to create the entire bacteriophage, and then it's going to release. And by all releasing methods here, it's always going to be lytic and burst open its bacterial host. Okay? So let's just look at this from another picture, going through the process one more time with the bacteriophage. So again, the host cell is down here in the orange with the purple being its genetic material. Here's the bacteriophage. So the first step is you have to attach. So here you see that it has attached to the host cell. The second step is it has to penetrate, and so it's taking its green DNA and it's inserting it into the host bacterial cell. So here's the green DNA inserted into the host cell. Now, at this step, it has to make a decision. Is it going to be active and go through the lytic cycle, or is it going to go into the latent phase and it's just going to incorporate itself into the host DNA? If it goes into the lysogenic phase, notice every time the bacteria replicates, it's going to make that viral DNA incorporated into its DNA, and it's just going to keep making it. Ultimately, under stressful conditions, the lysogenic bacteria, ultimately that DNA from the virus is going to pop out and force it up here into the lytic active stage. And then here, this is steps 3 and 4, where you're duplicating the viral proteins and you're synthesizing the viral DNA, and then you're going to be going through the maturation processes, and then once it's all assembled and matured, then through lytic, or bursting open, it forces itself out of the host cell, and then it goes and spreads on to further infect more and more and more bacterial cells. So again, that is the life cycle of an animal virus and of a bacteria virus. You need to know the steps, and then you kind of need to be able to compare those. So is this step occurring in both bacteriophage and the viral and the viruses that infect animal cells, or is it just one or the other? If you have any questions, don't hesitate to call me.